Here's another example of a manipulation of our body fluid compartments. It's really not so much a manipulation, but a disturbance, I should say, of the body fluid compartments. Look at this poor little girl. She lives in a country where she is economically disadvantaged. She does not have all of the essential amino acids in her foods. She's living on a corn-derived diet, and corn does not have tryptophan, which means we're going to have a problem with other amino acids and protein synthesis. So she has Kwashiorkor, which means disease of deposed child, or displaced. I've read it like that as well. It means that her mother has had another child, possibly, and this child that you see in the picture is no longer receiving breast milk. And the breast milk from her mother would have all of her essential amino acids. So in essence, she's no longer being suckled. And now she's on solid foods. She's not getting all of her amino acids. Well, how does this relate to a change in body fluid compartments? Notice her very distended belly. This is not from her being fed a lot of food. This is not obesity. What happens is this, and you should really be mindful of this. I go after this on an exam heavily. And this is an issue we will see throughout the entire semester. The liver does over 500 functions, one of which is to synthesize albumin. Albumin is the most abundant protein in the plasma compartment. It's about 7 grams percent, roughly. The 7 grams percent is the percent of protein in the plasma compartment, and most of that comes from albumin. So her liver is not getting all of the essential amino acids it needs in order to make this albumin. That means albumin quantities in her plasma decrease. That means the osmotic pressure in her plasma compartment is no longer 300 milliosmol. Let's say, just for discussion purposes, it drops down to 250 milliosmol. Now that means all of her other body fluid compartments are still at 300, while her plasma compartment is 250. There is a cavity in the peritoneal region called the peritoneal cavity. It is filled with peritoneal fluid. This peritoneal fluid in the peritoneal cavity is surrounded by epithelial tissue. That means it's a transcellular cavity or transcellular body fluid compartment. So her plasma fluid will shift, the water will shift from plasma into the peritoneal cavity. And that's why you're seeing her stomach so distended. That's edema. Specifically, when we find this edema in the peritoneal cavity, it's called ascites. So we see this not only in underdeveloped countries, but we can also see it in hospitals and nursing homes, where maybe you're dealing with patients who are really picky about what they eat, and they don't eat a well-rounded meal. So you can absolutely see this in other places besides third world countries. You can also definitely see this in alcoholics. As their liver fails, they cannot make albumin. Their hepatocytes are replaced by scar tissue. Cirrhosis of the liver is what that's called. And they don't make albumin. And then they get the same edema/ascites in their peritoneal cavity. So all of this water shifting is now from low plasma protein. Pay attention here. In the previous slide, we saw plasma osmolality climb and water shifting with the diabetes example. Now we're seeing plasma osmolality decrease and shifting of water from the plasma compartment into surrounding adjacent compartments. Also, look how swollen her feet are. That's for more gravitational pull of blood down to her feet, and more of that hydrostatic pressure plus a lower osmotic pressure of the plasma compartment leads to even more shifting of water out into the interstitial space in her feet. You can also see the wasting of her musculature in her arms and legs. That's her skeletal muscle trying to help in this situation by mobilizing proteins in muscle to send amino acids to the liver, trying to help boost the albumin levels. But again, as we saw in the previous slide, it's a losing battle. Our book refers to tonicity and osmolality as the same word. Are they really interchangeable? And the answer is absolutely not. Well, if they're not interchangeable, why does our book do this? Why does Guyton and Hall Medical Physiology seem to talk like they are interchangeable? Let me explain. Throughout this PowerPoint, I have been talking about osmolality. Osmolality refers to the number of particles per unit volume. It does not tell you anything about the permeability of those solutes. If you notice, in every slide where we had the manometer tube with the red solutes that could either move because they were permeable or couldn't move because they were impermeable, I had to tell you whether or not they were permeable. That was an extra sentence I had to say. If I simply say a solution is hyperosmotic, all that means is it has more particles per unit volume on one side of the biological membrane, the outside, compared to the intracellular fluid. That's it. It does not say anything about permeability. Therefore, if I tell you your cheek cell is placed in a hyperosmotic solution, and then I ask you, what will the water do? Which way will it shift? You actually do not have enough information to answer that question. I have to tell you whether or not the solutes are freely permeable. A hyperosmotic solution may cause only transient shifts in the body under steady state conditions, meaning the solutes, given enough time, are going to be permeable and will reach an equilibrium. So osmolality, that term by itself, does not tell you if the solute is an effective, which means it's not permeable, impermeate, or ineffective, which means it's permeable. I have to tell you that. But if I say tonicity, this actually is a word that tells you two things. If I say a cell is placed in a hypertonic solution, this now tells you two things right off the bat. One, the cell is placed in a solution that has a higher concentration of solutes per unit of volume than what's found inside the cell. Number two, it also tells you that these solutes that are found in higher concentration are impermeable. That means they are effective osmoles. I don't have to add another sentence. I can immediately ask you, will water shift? And if so, which way? If I say a cell is put in a hypotonic solution, you can see here for letter C, that means the solution has a lower concentration than compared to inside the cell, and those solutes are impermeable. That means the water will shift into the cells, where there's a higher solute concentration, and the cell will swell. Shown here in letter B, to go back to a hypertonic example, you can see the solutes in the surrounding solution are more concentrated than inside the cell. If I say tonic, hypertonic, that means they are impermeable, and that means the cell will shrivel or crenate and lose water to the surrounding environment. Let's practice this concept. Here I have a red blood cell shown on the right hand of your screen. I'm going to put it into a 300 milliosmolal solution of sucrose. Sucrose is not permeable across the cell membrane. It's a 300 milliosmolal solution, and that means it's isoosmotic. But is it also isotonic? And the answer is going to be, what do you predict? Will the cell shrink or swell? And the answer is, this solution is not only isoosmotic, but it's isotonic. There will be no net change in cell volume. Its osmolality is also its effective osmolality, meaning it's isotonic. What about this example? What if I put the same red blood cell in a 600 milliosmolal sucrose solution? Well, clearly we already know it's hyperosmotic. We also know sucrose is impermeable, and that means it is also hypertonic. What will the cell do? Will it gain water or lose? It's going to lose water to the surrounding solution, and that means the cell will shrink or crenate. What about urea? Urea is actually permeable across most biological membranes, especially red blood cells. So if I put this red blood cell in a hyperosmotic solution of urea, what do you think will happen? As you saw in the previous slide, a hypertonic solution caused the cell to shrink. Do you think this cell will shrink as shown in the chamber? Do you think it will crenate? Well, let's do this. I put the red blood cell inside this hyperosmotic urea solution, but because urea is permeable, there will be no net gain or loss of fluid across the biological membrane. The red blood cell will stay the same size. In other words, the solution is isotonic long term. That's the end of this section for our body fluid compartments. In class, we will continue on during your lab time with a review on the metric system, how to convert from scientific notation to standard notation and vice versa, and how to calculate osmolality from a grams percent solution. And we will also learn how to go from an osmolal solution back to a grams percent.